Hello. We'll continue now, today talking about the pancreatic hormones by talking about the second most important hormone secreted by the pancreas, which is the glucagon. So glucagon is secreted from the alpha cells of islets of Langerhans of pancreas, and it is also secreted from A cells of the stomach and L cells of the intestine. Glucagon is primarily catabolic, used for the mobilization of stored energy. And its secretion is controlled mainly by the glucose and amino acid levels in the blood. Any increase in the glucose levels in the blood, any decrease in the glucose level in the blood will promote the secretion of glucagon. When blood sugar level decrease below 80 milligram per deciliter of blood, the alpha cells of islets of Langerhans stimulated, of course, to secrete more glucagon, which in turn increases the blood sugar level. On the other hand, when the blood glucose level increase, alpha cells are inhibited and the secretion of glucagon decreases. Glucagon is a polypeptide with a molecular weight around 3 kilodalton. It contains around 29. It contains 29 amino acids, and the half-life is around from 3 to 6 minutes. So how glucagon is synthesized? It is synthesized from, of course, the pre-pro hormone precursors called the pre-pro-glucagon in alpha cells, and then is converted to pro-glucagon, which gives rise to glucagon. About 30% of glucagon is degraded in liver and 20% in kidney. And the cleaved glucagon fragments are excreted through urine. So you can see that 50% of the circulating glucagon is degraded in the blood itself by enzymes such as serine and cysteine proteases. And you can see here how the plasma concentration of the glucagon is inversely proportionally with the blood glucose level. You can see that the blood glucose level decreases with increasing concentration, decreases with the decreasing concentration of the plasma glucagon. So how is action? What is the action of glucagon? Actually, it increases a blood glucose level. It uses a peripheral utilization of lipids and as well conversion of proteins into glucose. The action on carbohydrates, glucagon, of course, increases blood glucose level by increasing the process of glycogenolysis in liver that increases BSL within minutes, because you know the action of the glucagon around from 3 to 6 minutes. It releases glucose from liver cells into the blood, and as well it don't induce the glycogenolysis in muscles. Glucagon increases the gluconeogenesis in liver by activating the enzymes that convert the pyruvate into phosphophenol pyruvate. This increase and as well increase the transport of amino acid into the liver to be utilized for glucose formation. Of course, when the amino acid is existing in the blood. For glycogenolysis, glucagon increases the blood glucose level by the glycogenolysis by the following complex cascade of events. On the target cells, mostly liver cells, glucagon bind with the receptor and activate adenyl cyclase via the famous G protein. The glucagon receptor is a peptide with a molecular weight around 62 kD. This causes, of course, a cyclic changes in the activation of the formation of the cyclic adenosine monophosphate (CMP). This will activate protein kinase A. So basically, it's the same mechanism that convert phosphorylase B into phosphorylase A. This will promote the degradation of glycogen into glucose 1-phosphate, which is then dephosphorylated and the glucose is released from the liver. So basically, it is the opposite mechanism of storing of glucose. So you can see here that glycogen is converted into glucose 1-phosphate by the action of the glycogen phosphorylase, and this is have a counter or opposite action by the effect of glycogen synthase. So let's see here the mechanism by this diagrammatic sketch. So you can see here that the glucagon binds to the receptor. Once the glucagon bound to the receptor, it will activate the G protein, as you can see. This will activate, once the G protein is activated, the adenylate cyclase will be activated and will convert and will form the cyclic AMP in the cytoplasm. This cyclic AMP will activate the pKa, which in turn will activate the phosphorylase kinase, converting the phosphorylase B into phosphorylase A that promote the process of glycogenolysis. As well, pKa will activate the BGC1 and the PEPCK and the glucose 6-phosphatase, and this will promote the gluconeogenesis. You can see as well that phospholipase C, as you remember, its main action is to break down the diphosphate into IP3, and if you remember that the IP3 is an important messenger in increasing the amount of calcium in the cytoplasm. So you can see these, all these events will lead to the increase in the process of gluconeogenesis, decrease in the process of glycolysis and the glycogenesis. So basically all of this will add up to increase the amount of glucose that present in the blood. Of course, this will lead as well to increase the process of ketogenesis as well as the uptake of amino acid. So what about the action of the glucagon on protein? So the increase in amino acid level in blood stimulates the secretion of glucagon. Glucagon increases the transport of amino acid into the liver cells. And of course, these amino acids are utilized for the process of the formation of glucose from amino acid gluconeogenesis. While the action on the fat metabolism, glucagon increases lipolysis by increasing the release of free fatty acids from adipose tissue and making them available for the peripheral utilization. And this lipolytic activity of glucagon in turns, as we said, promote ketogenesis in liver. In the adipocyte, the glucose activates the famous hormone sensitive lipase, which breaks down the triglyceride or stored fat into diacylglycerol and free fatty acid, and this will be released into the circulation. The glycerol released into the circulation can be utilized by the liver for gluconeogenesis. And these free acids can be used by fuel by most of the tissues, mostly by skeletal muscles and liver. In the liver, the free fatty acid undergo beta oxidation and the conversion into ketone bodies. Glucagon as well have other action, including inhibiting the secretion of gastric juice, as well as increasing the secretion of bile from liver. There are some other factors that increase and decrease glucagon. For increasing glucagon secretion, exercise, low blood sugar level, amino acid ingestion, stress, and epinephrine. And of course, all of these action, the exercise and the stress and epinephrine, lead to consuming of your glycogen stores. So it's obvious that you need more glucagon to have a constant pool of sugar in your blood. Gastrin, a peptide hormone that stimulates the secretion of gastric acid by the parietal cells of the stomach and as well aids in the gastric motility. And it is released by the G cells in the pyloric atrium of the stomach and duodenum and pancreas. So these gastrin aids in this action as well. While cholecystokinin, CCK, is a hormone which is secreted by cells in the duodenum and this stimulates the release of bile into the intestine. And once bile is increased as well, this will aid to increase the level of glucagon secretion. While cortisol, it's actually a steroid hormone produced by the zona fasciculata of the adrenal cortex. And as well, it is released in response to stress and low blood sugar level. For factors that decrease the glucagon secretion, a hormone secretion like somatostatin, insulin, of course, free fatty acids, and ketones. So here you can see how glucagon dominates in the fasting state while we fast. So you see here the dropping in the glucose level. Let's have a pointer. The dropping in the glucose level will promote the alpha cell of pancreas to increase its secretion of glucagon. This in turn will affect the liver to do the three processes: the glycogenolysis, the gluconeogenesis, and the ketones, formation of ketones, as long as there is a prolonged hypoglycemia. That's mean the blood sugar level are very low for a long time of period. All these processes will end up by increasing the plasma glucose level. And of course, you need this increased plasma glucose level in the fasting state for the use of the brain and the peripheral tissues. You can see as well that the decrease in the plasma glucose level will affect the beta cells of the pancreas, because it will inhibit the beta cells of pancreas to secrete insulin, so insulin levels will be decreased. And this, of course, will affect the muscles, which in turn will affect the muscles and adipose tissue that push up or give another effect on the liver by increasing the lactate and pyruvate amino acids and the fatty acids release. This will affect the liver to have the direct action to increase the plasma glucose level. Of course, as all the mechanism we took before, there is a negative feedback that will inhibit alpha cells once the plasma glucose level have increased to the normal level. There is a negative feedback start from the glucose itself to inhibit the alpha cells of pancreas to stop releasing or stop secreting the glucagon. And hence the mechanism is, or the process or the metabolism is balanced. So let's talk about a third hormone secreted by the pancreas, which is the somatostatin. So somatostatin is secreted from the hypothalamus as well. The D cells or delta cells mainly from delta cells in islets of Langerhans of pancreas, as well as D cells in the stomach, and as well as the upper part of small intestine. Somatostatin is a polypeptide. It's synthesized in two forms, namely somatostatin 14 and somatostatin 28, and basically 14 and 28 based to the number of amino acids of the peptides. Both forms have similar actions, while the half-life time of somatostatin is around 2 to 4 minutes. Somatostatin brings out its action through as well cyclic AMP. So too much somatostatin will lead to a condition which is called somatostatinoma, which lead to decrease in the insulin and causes diabetes. Somatostatin is degraded as well in liver and kidneys. What about the regulation, the action and the regulation of the somatostatin? Somatostatin acts within islets of Langerhans and inhibits both alpha and beta cells. So it can inhibit both glucagon and insulin. It decreases the motility of the stomach, duodenum, and gallbladder. And it reduces the secretion of the gastrointestinal hormone gastrin and CCK, as well as the GIP, gastric inhibitory polypeptide, and the vasoactive intestinal polypeptide. The hypothalamic somatostatin inhibit the secretion of growth hormone and thyroid stimulating hormone from the anterior pituitary. That's why it is also called growth hormone inhibitory hormone, GHIH. It's another name for the somatostatin because it inhibits mainly growth hormone. For its regulation, for the pancreatic somatostatin, the secretion of pancreatic somatostatin is stimulated by as well glucose, amino acid, and CCK. Any tumor in the D cells of islets of Langerhans causes, of course, a hypersecretion of somatostatin. This will lead to hyperglycemia and other symptoms of diabetes mellitus. For the gastrointestinal tract somatostatin, its secretion in the GI tract is increased by the presence of chyme containing glucose and proteins in the stomach and the small intestine. That's mean when you eat a meal that is rich in carbohydrates. For another hormone from the pancreas, it's called the pancreatic polypeptide, or PP. PP is secreted from the F cells or PP cells, gamma cells in the islets of Langerhans, and it's also found in the small intestine. PP is synthesized as well from the pre-pro hormone that is converted into actually it is a pre-pro pancreatic polypeptide in the PP cells of islets of Langerhans. It's a 36 amino acids and its half-life time is around 55 minutes. It suppresses, so PP actually suppresses the pancreatic secretion and stimulates the gastric secretion. And it is believed to increase the secretion of glucagon from the alpha cells of islets of Langerhans. So it brings the action as well through cyclic AMP, and it is degraded and removed mainly from the circulation by the kidneys. And its secretion is stimulated by the presence of chyme, as we said, chyme containing more and more proteins in the small intestine. So let's see here how the blood sugar level is regulated. So for regulation of the blood glucose level, as you can see here, the normal range is controlled mainly or majorly by the insulin and the glucagon. Insulin, of course, decrease the blood glucose level, while glucagon have the opposite action of increasing the blood glucose level. So in normal persons, the blood glucose level is controlled within a narrow range. In early morning, after overnight fasting, the BCL is between 70 to 110 milligram per deciliter of blood. After or between the first and second hour after a meal, it rises to a maximum of 140. And can be this level of glucose level can be brought back to normal by the end of the second hour after the meal. We are talking here in a normal regulation of blood glucose level. This regulating mechanism is operated through liver, as we know, and the muscles by mainly the influence of insulin and the glucagon. So insulin is the only hormone that reduces the BCL. So actually it is anti-diabetogenic hormone. The hormone which increases BCL, the diabetogenic hormone or anti-insulin hormones. That's the name of the other hormones that increase the BCL, like the glucagon. The role of the liver in the maintenance of the blood sugar level. As we know, we talked about liver and its major function in regulating these hormones or the blood sugar level. So liver actually serve as an important glucose buffer system. When blood glucose level increase after a meal, the excess glucose, as you know, is converted to glycogen and stored in the liver. Afterwards, when the blood glucose level falls, of course, it is needed to convert the glycogen stored in the liver into glucose to be released into the blood. And the storage of the glycogen and the release of glucose from liver mainly are regulated by the insulin and the glucagon. So the insulin decreases the BSL, and it is only, as we said, the anti-diabetic hormone available in the body. While the glucagon, as we said, increases the blood glucose level. As we said, there is a role of other hormones in the maintenance of the blood glucose level. The hormones that increases the blood glucose level are growth hormone, thyroxine, cortisol, adrenaline. Why it's important to regulate the blood glucose levels? Because glucose is the only nutrient that normally can be used by the brain, by the retina, by the germinal epithelium of the gonad. So it always needed to be in sufficient quantity to provide the optimum level of energy within these organs. So it is important to maintain the level of glucose concentration in a threshold that is high enough, that is necessary for nutrition. So here you can see how there is an integration of the energy metabolism. So the individual tissues do not function in isolation, but form a part of the community. And the need communication between tissues are mediated by the nervous system, any circulating substrates and the plasma hormone. So you can see from the figure, it is integration between different parts of the body to control the availability of hormone, the amount, the concentration of hormone. And this is coordinated by the nervous system according to the availability of the circulating substrate. So this is controlled mainly by the action of insulin, glucagon, epinephrine, and norepinephrine. Any change in the hormonal level allows the body to store energy when food is available. So let's here see how the effect of some of the hormones, or most of the hormones, on the pancreas itself. As you know, pancreas have different kinds of cells. So here you can see that the beta cells of the pancreas normally secrete insulin. That of course control the glucose concentration. The secretion of insulin here will inhibit the action of the alpha cells, which secrete glucagon. Actually, the alpha cells secreting hormone, as well, it will affect the beta cells of pancreas. You can see that the alpha cells normally secrete glucagon to activate the delta cells that can secrete somatostatin that inhibit the beta cells. As well as the delta cells can secrete somatostatin that inhibit the activity of the alpha cells. While the epsilon cells normally secrete ghrelin, which can inhibit both the alpha cells and beta cells. So basically, it can inhibit the secretion of insulin and glucagon. So basically, ghrelin is a hormone that produced and released mainly by the stomach, and small amount are released by small intestine, pancreas, and brain. And it is termed the hunger hormone because it stimulates appetite, so it can increase the food intake and promote fat storage. So this is actually a very good figure that shows how different hormones are coordinating to control the glucose homeostasis in our blood. So you can see here the low blood sugar level which reaches around 40 milligram per deciliter in blood. This mainly will affect the hypothalamic regulatory center, that will affect the pituitary gland, that lead to the increase the secretion of a the increase that will affect, of course, through the secretion of the ACTH, that will affect the adrenal gland, causing the release of cortisol and epinephrine, and as well as norepinephrine. As well, the effect of low blood glucose will directly affect the pancreas to secrete glucagon. This, of course, will lead to enhanced activity of the glycogenolysis and the enhanced activity of the gluconeogenesis. You can see how the different action of hormone will have their effect on a different mechanism. Of course, all of this will lead to increase of the amount of glucose, since as well that pancreas, the insulin secretion from the pancreas have been shut off. You can see as well here how the critical values for the different concentration of blood glucose levels. So you can see that blood glucose level, when it reaches around 50, this will let's talk initially from the beginning. It's around this concentration, you can see that the insulin production has been decreased. Once it got low, starting to go low in concentration of the blood, this will promote the action of the epinephrine, and the glucagon is increased. If the blood level still it's not critical because it's still in the blue zone. So if it goes, the concentration goes low again around 65 or something, the gross hormone production increase. If it goes around 60, you can see that the cortisol production start to increase. Once it reaches around 55, the adrenogenic symptoms begin and brings the anxiety and the sweating and palpitation. If it goes critically around 50, you can see that it starts, a lot of symptoms start to appear, even coma and death. So you can see it's a critical value that starts below the 50 or the 40 because the scale shows a red scale. So actually this figure is very good in showing the sequence of hormone that start to be secreted and increase in the blood sugar level, all of them aiming to rise the blood sugar level towards the normal range. Okay, let's see here the coordination of the mechanisms and the process to control the blood glucose level in the circulation. As you can see, it's mainly due to the effect of the insulin and glucagon, and this will control the feedback mechanism and the cycling of this concentration of glucose inside the blood. So initially, as you can see, that the action of the insulin is the Glucagon after eating a meal that increase the blood glucose level higher than the normal point. So actually, there is a set point in which this leads to the action of insulin by increasing the amount of secreted insulin from the pancreas, which causes the pancreas as well to release insulin in the bloodstream. As we said, it will affect the liver to begin breakdown, taking up the glucose and store it as a glycogen. This, of course, will lead to decrease in the glucose level and lead to normal glucose level. So this actually is like a negative feedback mechanism to the amount of the glucose inside the blood. While the other part of the cycle, actually the action of the glucagon starts when you miss a meal or you have a glucose level that is below normal and below the set point. This will trigger the pancreas, of course, to release glucagon into the bloodstream, which have the counter action against the insulin and signal the liver to begin converting the stored glycogen into glucose to get back, raise the blood sugar level to normal. So this is like the fast cycle to summarize the action of the insulin and the glucagon on the blood sugar level. Here as well, you can see the concentration of each hormone in relation to the glucose in our blood. So you can see here, once the glucose the glucose in blood is below normal level, you can see that there is a basal level of the of insulin, and the glucagon, it's a little bit higher because it needs to keep this normal level. So once there is an increase in the glucose amount due after a meal, it starts to shoot up the secretion of the insulin in the blood. And you can see that the amount of insulin existing in the blood is actually directly proportional to the amount to the glucose level in the blood. As well, you can see there is a counteraction for the glucagon once the insulin is in action. You can see here how the effect of increasing glucagon and decreasing insulin will affect the process of lipolysis, and hence will affect the process of free fatty acids release, and as well as the hepatic output of ketone bodies, which lead to ketoacidosis. So again, here we summarize the factor that affect the release or the secretion of insulin and glucagon release. So of course, the increase in plasma glucose will increase secretion of insulin and decrease the secretion of decrease the secretion of glucagon. The same happened for the and as well when increasing amino acids and the plasma GIP will increase insulin and increase glucagon as well. If the parasympathetic activity increased, this will lead to as well increase of both insulin and glucagon, while the increase on the sympathetic activity lead only to decrease the insulin and increasing of glucagon, because of course you need more glucose in the blood for more energy for the muscles. The increase as well plasma levels of epinephrine, which is the adrenaline, which will have the same effect of the sympathetic activity, leading to the increase of the secretion of glucagon. Here we'll summarize all of the hormones that involve in the process of homeostasis of the glucose. So you can see here, let's have a pointer. Okay, so you can see here that let's start from the pancreas. You can see that beta cells of pancreas is stimulated for the increased secretion of insulin and the increased insulin biosynthesis, obviously, and the increased beta cells proliferation, obviously, and increased beta cells in survival. So all of these four mechanisms all adds up for increasing the secretion of insulin. And actually, these increases of insulin are promoted or activated by the effect of GIP and GLP-1, which as well the GIP will affect the adipocytes to increase the process of lipolysis and increase the fatty acid synthesis. The GLP-1 can activate as well the beta cells and to decrease the amount of glucagon secreted from the alpha cells, and will activate the brain, of course you will have increased the nausea and decrease the food intake and decrease the body weight. This in case that there is not enough glucose is going to the brain. The GLP-1 as well will affect the stomach, and it's emptying of the stomach. That's moving the food will stay more in the stomach, will be decreased, and the stomach, as we said, increase secrete two hormones that aids in the glucose homeostasis, which is the gastrin and ghrelin. And this gastrin and the ghrelin will stimulate more insulin to be released through the formation of more beta cells, with the help of CCK coming from the intestine. Of course, as a negative feedback, this ghrelin can inhibit the secretion of the insulin, as you can see from the figure. So basically, this figure summarizes the action of a number of hormones on the glucose homeostasis. And this concludes our lecture today for the rest of the pancreatic hormones that control the blood sugar level.